Welcome to part one of our gastrointestinal topic. You'll notice there's an awful lot of independent learning with this one as well. Nothing too laborious, but it manages to give us a bit more breadth because there is so much to cover. There's also a collection of videos around cancer, about the cell cycle, that are extremely good. And they will help to underpin the underpinning that we're going to do today, because there's no point in me going through all of that when other people have done it so well. So today in this part, what we're going to do is we're going to look at the underpinning. First of all, we'll look a little bit at the structure of the ileum and the colon, just to sort of set some context. We'll look at GI immune regulation. These are the specifics on top of your normal immune inflammatory responses, but these are the specifics related to the GI tract. And as you can appreciate, that's quite complex because we have pathogenic and non-pathogenic microbes to deal with. We'll look at the causes of acute and chronic inflammation briefly, and then finally in this part we'll look at adenocarcinoma. And then in part two, we'll cover the applied pathophysiology. So let's do a bit of a comparison between the ileum and the colon. The ileum is involved primarily in the absorption of nutrients because, of course, it's first in line. That's where they hit, and that's where most of them are absorbed. And they're absorbed from that mulch, that liquid in the lumen called chyme. The ileum has little villi, little finger-like projections sticking up to increase the surface area. And the enterocytes within the villi have a brush border of cilia-like structures. Between the villi are some, if you like, indentations known as crypts. The colon absorbs water primarily in the proximal section. This, of course, helps to form the fecal masses, and then in the distal section, it stores them. It does have crypts, but it doesn't really have any proper villi. It also has many goblet cells because lubrication from mucus is quite important in the colon for obvious reasons. Both, of course, are lined by epithelial cells. These are simple, in other words, single layer, columnar epithelial cells known as enterocytes. Those enterocytes are joined by tight junctions, and this is what we've always said, that's a bit of a misnomer really, because these are selectively permeable tight junctions. In the ileum, the enterocytes at the top of the villi, villus singular, focus on absorption, but the ones in the crypts tend to be secretory. So those two areas have a different overall function. Secretory enterocytes in the crypts include enteroendocrine cells. These exocytose hormones. There's about 30 of them in the GI tract, and their role really is to regulate appetite, regulate absorption, and also motility. There's also some goblet cells, but in this case, unlike the respiratory tract, we don't have submucosal glands, it's just goblet cells. And we'll come back to this bit, patches of specialist immune cells. Blood and lymph vessels, not shown in the diagram here, extend up into the villi. Of course, they don't extend into the enterocytes because epithelial tissue, if you remember, is avascular. But they do extend up the villi. And in one respect, this is important because, of course, if we're absorbing all these nutrients, they then have to be carried away. And they're carried away to the liver via the hepatic portal system, other than fats, which are carried in the lymphatic system. So let's talk about that immune regulation. So GI immune mechanisms have a very fine balance, and we know that this goes wrong because this imbalance in the microbiota is being blamed for more and more things. But these immune mechanisms in the GI tract, they have to tolerate these non-noxious microbiota. This is actually a better term than microflora, and you'll notice it's tending to be used more these days. And this is made up of these commensal organisms. These are organisms that are not harmful and in some cases are helpful to us. And these make up an environment that's called the microbiome. So we need to tolerate that. But we also need to control for invading pathogens. So we need to be able to identify between, if you like the phrase, friendly bacteria, although we're not just talking bacteria, but we need to differentiate between our commensal organisms and invading pathogens so that we know which one to attack. If tolerance fails, of course, we end up quite often getting inflammatory bowel disease, and if some of the hype is to be believed, lots of other conditions. If protection fails, of course, we're going to start getting colonization of pathogens, and we're going to get a rip-roaring infection established. These commensal microbes start to be acquired by ingestion immediately after birth. And this then continues to develop right through childhood, which, if you remember, is when the thymus gland is at its most active, educating all the T cells. Remember, they have to go to school, and if they tolerate the right thing, then that's fine. If they tolerate the wrong thing, then that's not. They have to go and die. Remember this selection of T cells. The development of this microbiome also stimulates the development of specific GI immune mechanisms during human development. So if we inhibit the development of that microbiome, we might have a reduced or an abnormal response in terms of many of the immune mechanisms we're going to talk about. In adult life, as I said, these microbiota, these commensal organisms, are vital for the digestion and absorption of some nutrients. Some nutrients, we can't absorb them unless these commensal microbes alter them in some way and make us able to absorb them. And of course, like on the skin, they compete with pathogenic organisms and prevent the colonization of pathogenic organisms. By the time you get through that development stage, where this microbiome is created, it is unique to individuals. And by the time we get to adulthood, it is fairly stable. And as I've said, recent studies, they've linked sort of alterations in this microbiome, these levels of commensal bacteria, to various metabolic and inflammatory conditions. And the problem is, of course, in this case, is whether you can prove cause and effect. So are the alterations the cause, or are they an outcome? And this, as yet, isn't fully understood. But of course, it allows for the promotion of a load of probiotics and prebiotics. Because tolerance is developed pre-puberty, commensal microbes do not usually stimulate the adaptive immune response when presented to T cells, not in the way that we know, not in a pro-inflammatory way. So these microorganisms in the lumen have to be constantly sampled, and they're carried into gut-associated lymphoid tissue. You remember salt and malt. I think I did say at the time, we also have GALT. This forms Peyer's patches, little patches of lymphoid tissue below the enterocytes. You can see in the diagram on the right there. When they're carried off to the T cells, they don't stimulate the production of pro-inflammatory T cells. So we don't get, for example, T helper 1 cells. Instead, what we get are Treg cells, regulatory T cells. These Treg cells then go about their business, and they actually, instead of secreting interleukin 1, 6, 2, whatever, they secrete anti-inflammatory interleukin 10 and transforming growth factor beta. These are both anti-inflammatory. And by direct binding between the Treg cells and other cells, or via this secretion of interleukin 10 and TGF-beta, the Treg cells suppress the activation, proliferation, and secretion of T helper cells, cytotoxic T cells, B cells, and dendritic cells. So they help to inhibit the action of all the usual pro-inflammatory cells involved in the innate and the adaptive response. In terms of intestinal macrophages and dendritic cells, they also become less secretory of pro-inflammatory mediators, and they tend to secrete more interleukin 10, which, as we saw, is anti-inflammatory. Through all these mechanisms, what happens is you end up getting a general tolerance of the necessary commensal microorganisms. Which leaves us really with our pathogenic ones. We still have to deal with those. So, to try and resist those, we'll start again at the barrier level. We have the goblet cells. Goblet cells, of course, secrete mucin. Mucin then combines with water, remember the sol and then the gel layer, to produce mucus. Mucus lubricates and protects the epithelial barrier. It also, if you remember, contains pattern recognition receptors. Pattern recognition receptors, of course, combined to PAMPs and DAMPs. Mucus also contains antimicrobial enzymes. So it has some active immune response. Another thing we have in the GI tract are cells called Paneth cells. In the diagram there, you can see that little pink pyramid of cells in a crypt. These secrete defensins and other antimicrobial proteins. Defensins are a bit like membrane attack complex, except they don't form a little circle as such. They bind one each side and they create pores in pathogens. And just like our cells, that causes lysis of the pathogens and kills them. In inflammatory bowel conditions in the histology, the lab techs often see this metaplasia of the Paneth cells. So the reason they're pink in this diagram is because they take up a pink stain, so they put some pink stain in, and they notice that there's far more Paneth cells. because the existing enterocytes have then converted to become Paneth cells. This is metaplasia. Peyer's patches, as we said, are areas of this gut-associated lymphoid tissue, GALT, below the enterocytes. And the cells between the Peyer's patch and the lumen are characterized as an umbrella term as follicle-associated epithelia. In other words, the cells between the GALT and between the lumen. Some of these are these specialized microfold cells, or M cells. And these M cells constantly sample the microorganisms in the lumen of the GI tract. So they transcytose, that's endocytose, then exocytose at the base, both pathogenic and commensal microbes directly into the Peyer's patches. So it is just like a sampling process, a random sample is taken. And these then are passed through to the Peyer's patches, through the M cells. Of course, there are other areas of lymphoid tissue. There is still MALT, because we still have mucosa, and there are mesenteric lymph nodes. Intestinal dendritic cells are really funky because they can extend their dendrites. Remember, that's why they're called dendritic cells. They have all these little dendrites sticking out. They can extend the dendrites and phagocytose pathogens within the lumen. And this is called periscoping. So they can do it between the enterocytes, or they can do it through the M cells. So they can put up their dendrite, they can bind a microbe and draw it back down, either through the M cell or between the enterocytes. Of course, they're drawing it straight into lymphoid tissue. Another thing, I know we've talked about antibodies in the past, and we did mention IgA. We mentioned it in terms of it having these two monomers stuck together. So, of course, we've got 4 fragment variable sites sticking out. This is secretory IgA, slightly different. It's usually written SIGA, SIGA, if you like. It's continuously secreted by plasma cells. And of course, plasma cells are modified B cells. Our secretory IgA is transcytosed through the epithelial cells into the lumen at a rate of about 3 to 5 grams a day. And it does it by binding to the epithelial cells via its secretory component. If you look at the diagram, the secretory component is that yellow wiggly line running down, if you like, the stem of it. In the middle, we've got the joining chain. At either end, we've got our typical monomer with the heavy chain and the light chain. So it binds via the secretory component, and this stimulates the transcytosis of it through into the lumen of the GI tract. It can be antigen specific or it can be non-specific, and it has multiple functions. So specific secretory IgA, SIGA, binds via the FV ends, the fragment variable ends, to specific bacteria, viruses, or toxins. So this is more like a normal antibody action. This results in things like agglutination, and in this case we can agglutinate 4 targets at a time, entrapment in mucus, and all of that will then end up getting removed via peristalsis. If they get agglutinized, they get trapped in the mucus, they can't get in between or through the cells and colonize, and they're removed by peristalsis, and eventually, of course, they go down the toilet. Or this can result in the transcytosis back through the M cells. Non-specific SIG-A can bind via its secretory component again, that yellow wiggly line around it, S shape, to receptors, and these receptors are normally used by pathogens or toxins to get through or to bind to epithelial cells. So it can bind to those receptors that would otherwise be used by pathogens and toxins. And of course, this is neutralization. This blocks the pathogen or the toxin from binding and prevents pathogenic antigens from entering the cells. Pathogens and toxins, again, are then removed by peristalsis. Non-specific SIG-A also appears to bind via the secretory component again, because the non-specific actions all tend to be binding via the secretory component, and it binds to the commensal, the friendly bacteria and microbes. And what this seems to do is encourage the microbiome colonization. So what it's doing is it's building up and developing the commensal microbes to increase their function. And of course, specific and non-specific SIG-A is very prevalent in maternal milk. This is the immunological protection, or part of it, that you get from maternal milk. And then, of course, there's the tight junctions. And when we learned about the functions of tight junctions, this is one of the things that we spoke of. Of course, later on, when we talk about diarrhea, we're going to find out that they do allow the passage of water and ions. But they prevent the passage of microbes and large solutes. Microbes, of course, that stops them getting deeper into the tissues, where it's harder to kill them and get rid of them. In terms of large solutes, it prevents malabsorption because it forces solutes that we do want, nutritional solutes if you like, to be transported through the enterocytes and then metabolized so that we can use them. So normally they would prevent the passage of microbes. But some pathogens secrete toxins, particularly bacteria, although there is a virus that does it as well, that destroys the tight junctions. When this happens, of course, it means that the pathogen can get between the enterocytes and into the deeper tissues. So when we consider what possible immune mechanisms we might need, we have to factor in that they need to be able to get deeper into the tissues. So common pathogens, you have a handout and a quiz on this. If you haven't already looked at it, please do now. And in terms of the exam, but also in terms of your clinical practice, you need to be able to link those common pathogens and their usual mode of action, the things that they cause within the body, because that will tell you what signs and symptoms they're going to demonstrate, but it will also enable you to understand the immune mechanisms that are required from us in order to counteract them. So you need to know their mode of action, really. So in the handout we talked about, in terms of bacterial gastroenteritis, Campylobacter, Salmonella, Escherichia coli, E. coli, and Clostridium. In terms of viral gastroenteritis, we focus particularly on norovirus and rotavirus because those are the two most common. We looked very briefly at parasites as well. In terms of chronic inflammation, again, you have an independent learning handout on this. They're not massively dissimilar, Crohn's disease and ulcerative colitis, but it would be helpful if you had a comparison in your head between the two, whether it goes through the wall fully, partially through the wall, the areas commonly affected, that sort of thing. And also in that handout, we looked at diverticular disease and diverticulitis, slightly different from Crohn's and ulcerative colitis. So please see the inflammatory bowel conditions handout and the quiz. Which brings us on to adenocarcinoma. And again, you have some preparation for this. You have quite a few YouTube videos. Start with the cell cycle phases, because then that will lead you into what happens when that cell cycle goes wrong, particularly in terms of what Armando has called oncogenetics, where he talks about tumor suppressor genes, the brakes, and oncogenes, which are the accelerators. There's also a series of 8 short videos designed for medical students. They're not that bad, honestly, on cancer biology. And finally, in your videos, you've got Dr. Anthony Goodman. He's got 2 videos on there. They are quite lengthy compared to the others, but you will be fascinated by him, I think. He's quite amazing. He sits there in his lovely white coat with his stethoscope on his desk and looks so traditional. But they are really, really good talks. He really explains these things very well. So he's definitely worth a watch. And those links to all of those are available on Moodle. So in terms of adenocarcinoma, let's look at some of the key concepts that were covered in those videos. The normal cell cycle is driven by the products of proto-oncogenes. Don't forget, in this word we have the word gene. So we're talking about a series of DNA that codes for something, a protein. So hence we say the products of proto-oncogenes. So these are our accelerators. So our normal cell cycle, the most common accelerators involve the activation of this RAS protein, which gives us cyclins and CDK, which is a kinase, but also includes any mitogen. Mitogen is an umbrella term for a growth factor, or a component of the mitogenic mechanisms. We know that signaling cascades can be quite complex. So a proto-oncogene could code for a receptor or an enzyme within any of those cascades. And here's our breaks. The cell cycle is inhibited by the products of tumor suppressor genes, TSGs. This happens if there is any damage or if there's a lack of nutrients to enable the cell cycle to go forward. And the processes may involve a complete halt or even apoptosis, and this is stimulated by the products of gatekeeper genes. So in other words, we've got our gatekeeper genes, our P53 or retinoblastor gene, RB. A key gatekeeper gene in FAP, familial adenomatous polyposis, I can see why they call it FAP, is the APC gene. Don't need to know what it is in full. If we don't get to that stage, we can hopefully do some repair. And this is where caretaker genes come in. And common examples of the caretaker genes that affect repair are the breast cancer genes BRCA1, BRCA2, or another one called PARP, PARP1. These are caretaker genes. So they're trying to affect repair. If they can't, in come the gatekeeper genes and either halt the cell cycle completely until the repair can be effected, or send that cell into apoptosis so that the damage is not replicated. And cancer risk, if you remember, increases with both increased rate of cell proliferation. So cell proliferation on its own is not necessarily malignant. But what we do tend to have is decreased cell differentiation. This is where you start to have an increased proliferation because you're going through mitosis over and over and over and over again. There is increased chance of damage and mutation to other genes. That damage or mutation or changes to other genes will cause that cell to not function as it should. In other words, it starts to lose its normal function, and this is decreased cell differentiation. It's called anaplasia. So we can categorize malignant tumors as being fast growing, there's your cell proliferation, locally invasive, but also they tend to produce metastases, apart from in the central nervous system and in terms of basal cell carcinomas. All the others will eventually create metastases either at the next organ within the circulation or complex tissue with a capillary bed, or within a lymph node, a draining lymph node. So let's have a look at the abnormal cell cycle. If proto-oncogenes mutate, they are known as oncogenes. They then become more active. So they push the cell cycle harder. It's a bit like your accelerator has become stuck on. An example, and in one of your videos they talked about telomeres. Telomeres are end caps on chromosomes, and they tend to give the cell a limited lifespan because every time that cell replicates, those telomeres shorten until after about 50 to 70 cell divisions, you reach this point that's been called the Hayflick limit. Once that's reached, the cell goes into apoptosis. And you can imagine this is really useful for keeping the cells free of damage, keeping them young and fit. It's, if you like, it's euthanasia of cells. The enzyme telomerase replaces the lost telomeres. And in some cases, this is really useful. It's useful where we have highly replicative cells, so cells that proliferate easily, that we want to proliferate easily, so thinking epithelium, thinking epithelial cells of the liver. But it does in other tissues result in aging cells. Those aging cells become prone to damage. And most adult cells don't normally produce telomerase for that reason. However, the proto-oncogene, HTERT, you don't need to know what it stands for, I'll tell you the H stands for human, codes for telomerase. Now, if this proto-oncogene becomes an oncogene, then it's going to it's going to code and increase the amount of telomerase. This increased telomerase is going to protect the telomeres where we don't want it to. And this is going to stimulate unlimited cell replication. And 80 to 90% of tumours have this mutation. However, our tumour suppressor genes, if they mutate, they are ineffective at halting the cell cycle. So whereas our oncogenes are a bit like the accelerator stuck on, this is like our brakes failing. So if they mutate, they're ineffective at halting the cell cycle, perhaps initiating repairs if we're talking caretaker genes, or if we're talking gatekeeper genes, triggering apoptosis. An example of that are the viral genes expressed by HPV, human papillomavirus. These inactivate both of the major gatekeeper genes, P53 and PRB. And of course, in this case, we've lost both our gatekeepers, so we've lost our brakes. Or have we? Because that brings us on to the Knudsen two-hit hypothesis. In this, both copies of a tumour suppressor gene must be mutated or deleted, so completely taken away, for a complete loss of function and cancer development. Of course, we have 2 chromosomes. Each chromosome has the same tumour suppressor gene. So we must mutate and delete both to have a complete loss of function. And as I say, this is the Knudsen two-hit hypothesis. And what it really gives us is this inherited predisposition to a specific cancer. If you inherit the loss of one tumour suppressor gene, then you've only got one left. It's a bit like having one kidney. You need to look after the other kidney, because if you lose that one, then you're well and truly stuffed. So if you lose another in this case, then cancer will develop because you've taken out your brakes. So we have one mutated tumour suppressor gene, but the other one is still effective at putting the brakes on. So if we want to keep with the brake analogy, you can say that your handbrake has mutated, but you've still got your foot brake. However, only one proto-oncogene needs to be mutated, becoming an oncogene, because oncogenes are overactive. So here we have our pair of chromosomes. Each of the copies, allele, has a proto-oncogene each. In this case, if we mutate one, it becomes more active. So yes, the other one's still working. Of course, that's not helping the situation because we've got one that's gone really, really active. In other words, our accelerator is well and truly stuck on, and the only thing that can save us is some effective brakes. Of course, if we then start failing in the brake department because of increased proliferation and we get more mutations, and we start losing tumour suppressor genes in pairs, then we've lost our brakes as well. Most mutations are actually acquired rather than inherited, and they can be acquired very early, very soon after birth, rather than being inherited, and then ongoing throughout our lives, of course. So carcinogenesis is progressive. We can see that because we start with increased proliferation. That increased proliferation increases the chances that we are going to get more and more mutations, and we're going to get anaplasia, where those cells have poor differentiation, start to become invasive and end up metastasizing. So because of uncontrolled proliferation, where we have, say, for example, mutated oncogenes, there are increased odds of mutations happening to other parts of the DNA. Any damage or mutation will be replicated, particularly if we have mutated and ineffective tumor suppressor genes in this increased proliferation. So we end up with increased proliferation, loss of differentiation, invasion, and possibly metastasis. So what mechanisms contribute to this, make this even more likely to happen? First of all, there's self-sufficiency in growth signals. So this involves either the synthesis of its own growth factors by the tumor, or overexpression of growth factor receptors or alterations in the signaling pathway. There is also insensitivity to anti-growth signals, and often the RB tumor suppressor pathway is targeted at various levels to try and knock that out, to knock some of the brakes out. You remember contact inhibition. You remember we said that cells basically butt up to each other. If you remove one, it will make the adjacent cells go into mitosis to fill the gap. Once they feel each other next to each other again, that stops them and puts them into the rest phase, the G0 phase of the growth cycle. In this case, the mechanisms above override that and you lose contact inhibition. Evasion of apoptosis. So obviously this could happen if you lose P53, that's our gatekeeper gene that sends the damaged cell into apoptosis so it can't replicate, or it can involve the activation of something called BC12. BC12 is anti-apoptotic. It prevents apoptosis. So if you activate it, it's going to block apoptosis. Cancer cells are very sneaky. They tend to use necrosis rather than apoptosis wherever possible, because what they want to do is they want to stimulate the inflammatory response. If you remember necrosis, we have a much messier death of cells. When there's necrosis, we're more likely to recruit more inflammatory cells, secrete more inflammatory mediators, and some of those inflammatory mediators are going to be growth factors. They're going to be growth stimulating cytokines, because normally we would need those for repair. In this case, the tumor is using it to promote its own growth. And this is back to our telomeres. So if you remember, we said a large majority of mutations occur to HTERT. This becomes upregulated, which then upregulates telomerase, which then protects the telomeres. And instead of having cells that reach a certain lifespan and die, they carry on replicating beyond their normal lifespan. Senescence is lost. Tumors also want a good blood supply, so they express pro-angiogenic factors. These are factors like vascular endothelial growth factor, fibroblast growth factor that stimulate angiogenesis. And again, normally in a repair situation, that would be really helpful because it brings in nutrients for the healing tissue and repairs the blood supply. Remember those little capillaries budding off and coming into the new tissue. Of course, a tumour wants that to happen because it gives it nutrition and the ability to grow. There are also anti-angiogenic factors. You've got an example there, thrombospondin 1. So these are suppressed. So we lose the control of angiogenesis and promote angiogenesis itself. So we end up with new circulation, this neovascularization, which allows for increased growth, but also think about it, it allows for metastases, because what we're effectively doing is we're putting in a road for them to go out on. So we've got new capillaries growing into the tumour. If a malignant cell breaks off, then it will get into that blood supply and get carried off to the next tissue bed, the next capillary bed in the circulation. So it's a little route out for the malignant cells so they can metastasize. The tumour also uses the inflammatory process to break down the extracellular matrix. Of course, it wants to do this because what it wants to do is grow into it. And normally, when we're repairing tissue in a normal healthy situation, we need to do it as well. So we use those matrix metalloproteases, things like that, to break down the extracellular matrix of collagen that the fibroblast laid down so that we can proliferate useful cells into that area. Of course, in terms of a tumour, that's really helpful for it because it allows it to invade other tissues, invade the tissue itself, break through the basement membrane. It gives it space to grow. And as we said, as we've seen, it also promotes growth factors. So tissue invasion and metastasis, our cancer cells require the ability to invade into the basement membrane and detach from neighbouring cells. They have a new blood supply, and this allows dissemination to distant sites. Both invasion and angiogenesis allow this dissemination, either via blood vessels or via lymph. So colorectal cancer. Now, this one is often diagnosed only when it metastasizes. So metastasis, wherever the primary is, can cause many other signs and symptoms depending on where it metastasizes to. And often it's those signs and symptoms that then get picked up. Colon primaries generally metastasize to the liver because of the hepatic portal system. So they get into the hepatic portal system. The liver, as we'll find out when we do our next topic, has a very fine network of little sinusoid capillaries that run through the lobules. So it's a perfect place for any rogue cells, malignant cells, to get lodged. They get lodged in the microvasculature of the liver, even more so if there's a predisposing liver disease that's going to block off those little sinusoids and make them narrower. And we end up having a secondary in the liver. So metastases usually occur first in the capillary beds of the next organ within the circulatory system, because they travel there through nice wide vessels, get there, and you've got a capillary bed. Lots of tiny little capillaries. Very easy to get lodged in those. Or they occur in the draining lymph node if they get into the lymphatic system. Colorectal cancer usually starts as a benign polyp. So this is adenomatous polyp. And you can see here it'll start in the usual way, hyperproliferation, works its way up to dysplasia, where it starts to have significant changes in the function of the cells. We then go into full-blown adenocarcinoma, which ultimately will invade the basement membrane and finally metastasize. And of course, the risk of colon cancer is going to increase with the number of adenomatous polyps. And people that have FAP tend to have hundreds and thousands of polyps, so they will often develop colon cancer. If one polyp goes untreated, 4% of people after 5 years and 10% of people after 10 years will develop colon cancer. And of course, what happens is you get mutations in various proto-oncogenes and tumor suppressor genes because of this increased proliferation of the polyp that then turns it into a malignant adenocarcinoma. So that was the end of the underpinning part. You now need to move on to the applied part, where we talk about common signs and symptoms in GI conditions. If you haven't already, please complete all of the independent learning activities. There are some optional articles in a folder. Have a look at those. If they're relevant to your clinical practice or you're just interested, then feel free to have a browse. And do all of the usual things. Get each of the topics under your belt fully so that you understand them, making your own notes, you're talking about them with your study buddy, teach someone at work, those sorts of things. And just to remind you, we still have 2 more topics. We have our hepatic session and we have our neuro session. And just to give you a bit of a countdown, your final exam is in 10 weeks' time. So, on to applied.